Hey there, and welcome to the Simplified Hub platform. In today's explainer, we are tackling the complex, absolutely fascinating process of meiosis. Look, if you're a biology student trying to completely master this topic, you have landed in the exact right spot. We're going to build your knowledge up step by step. All right, really quickly, here is our study guide agenda. We'll start with the basic rules of the game, move chronologically through Phase 1 and Phase 2, compare those phases side by side, and then wrap the whole thing up with a quick practice quiz.
So let's jump right into Section 1, the basics of meiosis.
Before we get into the weeds, we absolutely have to define a crucial term, haploid. These are cells with exactly half the normal amount of genetic material. In humans and other animals, we're talking about your sex cells. Honestly, this definition right here is the entire point of meiosis. The goal is producing these specialized haploid daughter cells from one original parent cell. If you get what haploid means, you basically understand the big picture goal of this whole process. Right, so why does this matter? Well, meiosis guarantees that all organisms produced via sexual reproduction end up with the correct number of chromosomes. It literally does the math to ensure you get exactly half from each parent. Think about it. Without meiosis perfectly having that genetic material first, sexual reproduction simply wouldn't work. The math just wouldn't add up.
Let's move on to Section 2, Phase 1, the very first split.
There are 4 key stage names you need to memorize for this: prophase, metaphase, anaphase, and telophase. You're actually going to see these repeat later, so let's look at exactly what goes down in each one during this initial division. We kick things off with prophase 1. Here, the chromosomes condense and the nucleolus breaks down. But the real star of the show here is crossing over. As these chromosomes move around, they exchange genetic material. To really visualize this, imagine taking 2 different colored markers, like a red one and a blue one. If you rub their wet ink tips together so the colors kind of swap and bleed into each other, that is exactly how genetic material exchanges between chromosomes. It's this awesome mixing of traits right at the starting line. Next up, we have metaphase 1. This is where random assortment occurs, meaning those chromosome pairs line up completely randomly right down the center of the cell. Now, what is super critical to remember here is that they align in pairs, 2 by 2. Do not forget that detail, because it's absolutely vital for understanding metaphase 1.
Following that, we slide into anaphase 1. Because they were lined up in pairs just a second ago, it's the whole chromosome pairs that separate and get pulled to opposite ends of the cell. This specific movement is exactly what ensures each new cell gets half the number of chromosomes, achieving that haploid state we talked about at the very beginning of the explainer.
To wrap up the first half, we arrive at telophase 1. 2 new nuclei form around each set of chromosomes at the poles, and then the cytoplasm splits. The result? We get 2 haploid daughter cells. So, phase 1 officially ends here with 2 brand new haploid cells. But, as you probably guessed, the process isn't quite over yet. We have a second division to tackle.
Which brings us to Section 3, Phase 2, the second division.
This second phase exists to create 4 haploid sex cells, specifically sperm and egg cells, from the 2 cells we just finished making. So we begin again with prophase 2. Notice how the cycle just restarts in our 2 new cells simultaneously. The chromosomes condense once more, the nucleolus breaks down in both cells, and they are gearing up for the final split. Then we hit metaphase 2, and the chromosomes align in the center of the cell again. But wait, contrast this with phase 1. Here, they line up single file, not in pairs anymore, but single file. This is a real game changer, and it happens specifically to ensure the very next stage goes off without a hitch. And here is that next stage, anaphase 2. Because they lined up single file, this time it's the sister chromatids that separate and move to opposite ends of the cell. This separation is absolutely vital. It ensures that the resulting daughter cells keep exactly half the genetic material of the original parent, staying perfectly haploid. We've finally reached the end of the physical division with telophase 2. 4 new nuclei form around each separated set of chromosomes, the cytoplasm split, and boom, 4 haploid daughter cells are formed. This is the grand finale of meiosis. One original parent cell has successfully transformed into 4 distinct, fully functional haploid cells. Let's move to Section 4, comparing phase 1 and 2. To really lock this into your memory for your exams, let's highlight the core mechanical differences between the 2 splits. In metaphase, phase 1 aligns in pairs, while phase 2 aligns single file. Because of that, the anaphase separation is totally different. Phase 1 separates chromosome pairs, while phase 2 separates sister chromatids. And finally, the telophase result gives us 2 haploid daughter cells at the end of phase 1, but completely finishes the job with 4 haploid daughter cells at the end of phase 2.
All right, Section 5, Practice Quiz.
It is time to put your active recall to the test. Just based on the facts we covered today, here's your first question. During which specific stage do sister chromatids separate? Think about the difference between pairs separating and sister chromatids separating. If you said anaphase 2, you are absolutely correct. Great job.
Moving right along to question 2, in which stage does crossing over, the exchange of genetic material, occur? Try to think back to our wet marker analogy earlier in the explainer. When did those colors swap? That genetic exchange happens exclusively during prophase 1.
And for our final quiz question, what is the final cell count at the very end of telophase 2? Think about the grand finale of this whole biological process. The ultimate result of the entire meiosis cycle is exactly 4 haploid daughter cells. If you got that, you have officially grasped the core objective of meiosis.
Meiosis really is an incredible biological engine, successfully transforming one single cell into 4 unique haploid sex cells. As you continue reviewing your study guide, I want to leave you with a thought to ponder. Given the crossing over of traits and that completely random assortment of chromosomes we just talked about, how might this randomized process drive the vast, beautiful diversity of life we see around us every single day? Definitely something to think about. Thanks for joining me on the Med Simplified Hub platform, and keep learning.